I remember to press the record button this time, Sky. Sorry about that. I'll have to fix that later. Just waiting for a few more people to show up and then we'll get busy. Okay, a couple more minutes and then we'll get going. Just so people don't miss stuff. All right. I think that's long enough. Don't have many people here today, but that's not a problem because I hit record this time. I can't believe I forgot that. For the lecture, I'll have to record a lecture for that for today. Okay, so essentially what we're going to do today is shouldn't take too long, really, because we have most of this information already available in the handout. So let's log into Blackboard. On Blackboard, if you go to the course content folder, which says should be flies or fly linkage, something like that. Oh. There you go, linkage project. There's not a lot in there. It's just a document, but that will give you all the information and hopefully the guidance necessary to do this lab. So let's bring that up. Now, here we go. Okay, get the chat open. All right, so actually, let's put this onto that. Cool. So essentially what we're doing in this lab, so it's going to be a series of 3 labs. There's a few different assignments as part of it, and this is a reasonable chunk of your lab grade as well. It should be fairly straightforward to do, and there's some really good instructions from Dr. Page on the graph making, but we can go through the basis of it now. So essentially this is using flies to calculate linkage, right? And so that's what we'd spend a lot of time doing today in terms of the nuts and bolts of it, right? The theory. The data that you'll be using here are actually generated from a actual cross between flies to be able to calculate the linkage distance between 2 genes. And we use flies because they're kind of neat. They're also really annoying, actually, flying all over the place. But they have some really neat observable phenotypes, and you can link that quite nicely to genotypes. So essentially, this genotype to phenotype link is what we're going to be working on for the last probably half of the lab in the module 1 to 6 labs, which are going to start, when's that? Thursday Tuesday the 7th July. So what's that? 2 weeks from now, we'll start that module. And there's a series of 6 modules which are all on Blackboard, basically working through linking the phenotype that we're going to talk about today, or one of them, with the genotype and doing some molecular and bioinformatics work on that. So let's get back here. So these are actually white eyes. So here we go. This is what white-eyed flies look like. They literally have white eyes because they don't have a pigment being produced in their eyeballs because they have simple compound eyes, which are pretty neat. So they have lots and lots of little lenses which create their vision. Anyway, so this gene was the very first that was discovered in flies by Thomas Hunt Morgan. Right, so this was the starting point for his experiments, which kind of led us to what we have today, trying to map all of the genes on the X chromosome of flies, and white is on the X chromosome. And so you're going to be working with 2 different traits, and I truly do wish you were able to do this in person, because it's a lot more context essentially. But what we're going to do is, at least on computer, do a cross between traits with 2 sets of flies which are either heterozygous females for this trait, and where are we at? This trait down here. So these are yellow-bodied flies. That's another type of pigment gene. Right, so you can see normally they're kind of brown, red eyes, obviously, because they're wild type. And the yellow-bodied mutants are a lot paler. And this yellow gene is what we're going to be working on later on for the molecular and bioinformatics approach to figuring out why they are the way they are, essentially. So there are 2 genes, white and yellow. So they're given the names of the recessive phenotypes, typically. So white and yellow are on the X chromosome, and your job is to try and figure out how far apart they are. So to do that, we're going to do a back cross. And so if you remember from the crosses that we did in terms of linkage today in the examples in the PowerPoint, what you do for these is you generate a heterozygote female, right? That's just what you want. And so in flies, we've got, so this is on the X chromosome, which is why you have a big Y over here. These on the right are males. They're hemizygous, so we have one X chromosome and a Y chromosome underneath it. And remember, if you go back to what we talked about today in a lecture, this is I find a more straightforward annotation to writing genotypes, right, for these kinds of linkage crosses. So we have a recessive male crossed with a wild type female, which is XX, so it has the two crosses for both, pardon me, both chromosomes. This will generate two types of offspring. It'll either generate wild type males, which normally we just kill them. Seems a little harsh, but I mean, I guess flies, they're going to die anyway at some point. And also heterozygous females. So what linkage phase are these females in? Another question. Just make sure you're all there. Yeah, they're in the cis phase, right? Doesn't truly matter because in terms of the output in some senses, but it's good to know because then you can figure out which ones are the parentals. You can also figure that out from numbers as well. So anyway, this is going to generate in the F1 generation two types of flies, and these are the ones we want. We want the females. And so we're going to take those females, right, which you see here, and we're going to cross them back, that's why it's called a back cross, to the hemizygous recessive males. And I keep saying hemizygous. Hemizygous means they only have one copy of each allele because they only have one X chromosome. You don't see that for autosomes. It's either homozygous or heterozygous. Hemizygous is for sex chromosomes where you have this sex chromosome and Y or W chromosome. Okay, so what that's going to produce, and really you got to follow the chromosomes here. So every male is going to contribute either a wild type X chromosome or a Y chromosome. Oh, I'm sorry, Angela, probably 5 minutes or so. We're just going to go over, well, we are just started going over, that's all right. Thing is, I don't want to wait too long because I don't know if people just aren't going to show up. So essentially what we're doing is we're going over the cross that we're going to be doing to investigate linkage between two loci on a fly X chromosome. One locus is the white gene, which when mutated gives you white eyes. So typically genes are named after the mutant phenotype. And the yellow gene, which gives you yellow-bodied flies when it's mutated. And so we're going to be doing, and this is really exactly the same as the linkage crosses that we were working through today in the lecture. And also, don't worry, Andrew, if you want to just go back and check, hopefully, fingers crossed, WebEx is going to work properly this time. And I actually started recording it from the beginning, which also helps. So you can always, I'll post this to YouTube, probably half hour, an hour after finishing this, kind of time it takes to convert. And then you'll be able to see what you missed as well. So anyway, the basis for a linkage cross is that you want to generate heterozygous females, or even, actually, it could be heterozygous males if it's an autosome, it doesn't really matter. In this case, you want heterozygous females because we're dealing with a sex chromosome. And so you can only have recombination between two homologous chromosomes, right? So you have to have one having one allele combination and the other one having the opposite allele combination. It's the only way you're going to see recombinants. And so this heterozygote or heterozygous female, and you can tell it's female because it doesn't have a big Y underneath it, is what we're going to use in the second cross. And this is the back cross. So this is going, essentially it's called a back cross because you cross it back to one of the parents. It doesn't actually have to be one of the parents because they might be dead by then, but it's to the same genotype that one of the parents has. And you can see I've also used another style of notation up here for writing the genotypes as well. So use whichever you feel most comfortable with, really, at the end of the day, that you understand best. Okay, so where were we? Right, so we're going to cross this heterozygote to anything that's recessive, right? It could be a homozygous recessive individual if it's an autosomal or autosome chromosome. And you can use your fingers here. I can't because I can't show you what I'm pointing at with my fingers. But essentially you have to track chromosomes. So the only chromosomes that the male can contribute to the next generation is either a Y, which will make a male, or a recessive chromosome, which is WY, right? Either way, it doesn't actually matter whether you score males or females or just combine them all together. What you're doing is you're providing like a blank slate, essentially, to see what chromosomes you get out of the female. So because these are standard dominant recessive traits, with the recessive chromosome, all you will see is the allele combination on the chromosome coming from the other parent. Does that make sense? And you can kind of see it down here. Every single one of these females will have the recessive chromosome from the male. So essentially, it's kind of doesn't exist, basically, in terms of phenotypes at least. Yeah, all of these are female. You can do the same again with males. Just replace all these chromosomes at the bottom with a Y. In terms of, because this is a sex-linked trait, it's on the X chromosome. In terms of the phenotypes you see, the effect is exactly the same. So if you kind of put your hand like so, really by putting your hand there, that's the contribution from the male. Everything above that, XX, W, or plus plus, W plus, plus Y, WY, that's all from the female parent. Sorry, I got a fan on which is kind of blowing in my mic. So it's really a blank slate upon which you can look at the chromosomes that come from the mother, because that's where recombination is going to be happening. So for example, you can look, the female can contribute it could contribute a plus plus chromosome, and so that will give you a plus plus over YW down here, right to the bottom left. Phenotype would be wild type. Hey Peyton, were you having problems joining the meeting? It's just because someone else had problems earlier. I don't know if we're having issues with WebEx or not. Oh, okay. Hey, so everybody, there's not an awful lot of people here, but I'll send out an announcement. Probably got to be Monday because I'm actually going to be away from the weekend, trawling around a swamp. Um, yeah, sorry about that, Peyton. WebEx is steaming pile of turd as far as I'm concerned. So I'm going to try and transition us over to Blackboard Collaborate, which, as far as I'm aware, isn't a steaming pile of turd like WebEx is. And basically, I'm only using WebEx because that's what I was kind of forced to use by the university. I was using Zoom before, which was much better, but they don't like it. Um, so yeah, the issues with the recordings, not this morning, that was my fault, but with the last lab where it's not synced properly. Um, yeah, yeah, I know, Angela, and that's just my own stupidity, to be honest. I went with WebEx because A, I was told to, and B, supposedly it works and it's supported. But that doesn't seem to actually make the slightest bit of difference in how it functions. So I should have listened to my friend who said, Matt, use Blackboard Collaborate. It's awesome. And I was like, oh, I better use WebEx because that's what I'm being told to use. So I should listen to him next time. Anyway, I've been thinking about that for a little while, but the last couple of issues have kind of tipped me over the edge on that. But I'll have to figure that out on Monday and let everybody know. Anyway, so yeah, sorry about that. At least it has been recorded, so fingers crossed WebEx is not going to screw up the recording and it'll make some sense. So yeah, sorry Peyton that you joined late. Um, Essentially, we're doing a back cross linkage experiment. And so this is the second stage of the back cross where we have a heterozygous female in this case, and we're crossing it back to a recessive male. And so these here are shown are females, but they could also be males too. It doesn't actually matter because what you want to do of all, right, which is the whole point of how this cross is structured, is you want to see whether or not the chromosomes coming from the female are recombinant or parental. If they're parental, you'll get the same phenotypes as in the parents, right? So this parent is double wild type, so it's red-eyed, brown-bodied, and the pictures are further up in the handout there. They're all there. And this male is white-eyed, yellow-bodied. And so the parentals, which is both the parental chromosomes and also the parental phenotypes, are red-eyed, brown-bodied, so wild type, and white-eyed, yellow-bodied, so recessive, right? So by doing the cross this particular way, really all you're looking at is the phenotype from one chromosome, if that makes sense. The other chromosome is not there effectively. And so what you're actually interested in, this is what we're going to be using, how many of those flies have a new combination of phenotypes from their parents. So that's white-eyed, yellow-bodied for this one, red-eyed, white-eyed, brown-bodied for this one, red-eyed, yellow-bodied for this one. So this is a combination that you do not see in the parents. And those are the result of recombination between these two homologous chromosomes up here. So just to recap, the first part of the cross is to generate those heterozygous females. Males, they actually go in what's called the fly morgue, which is where flies go to die. It's a little bit macabre, but is what it is. And so we then take those heterozygous females, which when they undergo meiosis or when they produce gametes, may or may not have recombination occurring between these two loci. And so we want to see whether or not that has happened. If it has not happened, we'll see the parental chromosomes and phenotype combinations that we see in the parents up here. If we do have recombination, now we're going to see a new mix, right, of, again, chromosomes and phenotype combinations. Okay, so essentially, and this is really my best effort to try and kind of recreate what you would be doing, but to do it online. And so, although you don't get the benefit of making fly media and chasing flies around over the lab and stuff like that, hopefully you'll still get the benefit of doing the analysis, right? Because those are the skills that I really want you to get. And so these data are from, I think these are collated from a bunch of crosses that have been done over the years, because there's a lot of flies there. And we've got the key here too, but you can actually figure this out because which of these combinations or classes are the recombinants? Give you a quick test. You could either work it out by the phenotypes or by the numbers. Which ones are the recombinants or the result of recombination? Exactly, because they have the smallest number. Right, so they're the red-eyed, yellow-bodied, or the white-eyed wild-type body, or red or brown-bodied. Right, so these ones over here are very rare. Gabrielle, we missed all the fun. We're having a party. Yeah, or not, actually. And so this is a very extreme example of linkage. So would you say that these are only slightly linked or that they're very tightly linked, these two genes? You can also compare that to some of the crosses that we did today in lecture. That's right, the two genes, so the white and the yellow genes. Yeah, they're really tightly linked because there is a vast excess of parents or parentals, right? So you look over on the left, we've got shit. I mean, for even just one sex, it's going to be 1,000 to 8, right? And so there is very little recombination going on between these two genes there. I'm trying to think of a appropriate I can't. Anyway, they're very tightly linked, right? Like brothers from another mother. There you go. That's kind of the best I've got. That it's not going to annoy anybody. They're super close, right? And actually, you can go look at the Drosophila genome, and you can actually look at linkage maps, and they'll show you they're very right next to each other, right at the end of one arm of the X chromosome, from what I remember. Okay, so that's the setup, right? So we've got these crosses, and these again would be the crosses that you would do yourself. And so essentially what I've done is I've laid out what you would be doing, and I've given you the data that you would have gotten, right? Just without you having to go in and stare down the microscope at flies a lot, right? Which, depending on your perspective, could be a good thing or a bad thing. Okay, so essentially what I want from you lot, and this is what we're kind of going through today. We'll go over bits of it next week too, as necessary, and it's kind of like a open forum troubleshooting type deal. Is I want you to present these data in a nice graph, right? Because even though these data, are fairly clear, it's still hard to interpret it just by looking at the raw numbers. And so what I want you to do is basically take these data and then watch this. Let's see if I can. Yeah, I thought that was a hyperlink. Oh, here we go. So anyway, this is what Dr. Page looks like. Handsome chap. And he's got a lovely southern accent to listen to. There you go. That's a little bit of a lie. It's 21 minutes long, but it's not too bad. Let's get that off. Stick that over there and get this back. Here we go. Getting there. And so what he'll do is he'll go through essentially the process of drawing that graph. And this is just a neat thing to do, right? It's a good practice. Some of you are already really quite good at this, which is great. It shouldn't take you too long. Others may have had less experience, and so this is really handy to just get you used to doing that. And really, in terms of biology, you don't want to present data just as a table. Sometimes it's handy. Most of the time, you want to be presenting it graphically because it's a lot more accessible, I think. The next thing that you'll need to do, and we'll go over this next lab on Tuesday, is because I need to re-record the lecture from this morning as well, so I want to get on with that before it gets too late. You're going to be doing a statistical analysis, a chi-squared analysis of these data. Right, and this is actually laid out very nicely for you. The data here are not your data that are being used here, but they'll do the same job, right, in terms of practice. And so what you need to do is take these data, calculate totals, calculate expected values from these, and then do a chi-squared test to see whether or not the data you get are what you expect. And so this is always a slightly tricky thing because essentially, how do you know what to expect, right? So you can take two extreme positions. You could expect that they are not linked. That's actually probably a better null hypothesis, to be honest. Or you could expect that they're completely linked, right? So there is no recombination between them. I'd suggest going with a more conventional null hypothesis that they are not linked, right? So essentially you would figure out, okay, what proportion of each combination of phenotypes, these four up here, would I expect to see if these genes were not linked? That's how you'll calculate your expected values, right? And we've got 8 different categories here. We've got four different phenotype combinations and 2 sexes. You could combine the sexes, then you only have 4 phenotype combinations. It's up to you, as long as you're clear about your workings. And then you just go through the standard chi-square goodness of fit test and see whether or not your observed data match what you would expect, right? And so you need to work out the chi-square value, degrees of freedom, and your p-value for those 2 things. You're also going to calculate an odds ratio, and so this is essentially the odds of having different options. And so what are the odds of having a white allele with a with one of these, with a wild type allele, wild type yellow allele, right? Essentially, if the odds ratio is 1, then it's an equal, there's no bias one way or another. So this is essentially looking at the probability of getting different chromosomes, right? And so that tells you how to do so down here. Got some funky formatting going on. Here we go, looks better. And then what I want you to do, oops, oh yeah, this is with the old data. I want you to calculate recombination frequency and map distance. Okay, so essentially what we've done today, but in terms of the lecture, but I want you to do a little bit more by presenting professionally the data, and that's going to be in Excel, doing an analysis of the data to see whether they fit what you would expect, and then finally calculating the distance between them. Okay, anybody have any questions so far? So you can see that even though you're not doing the physical work yourselves, you're still doing all of the analysis and the kind of intellectual work, which is the intent. And we'll be going over this part, the analysis and the graph making, next Tuesday, if anyone needs it. It's going to be, I'll be there, or technically here, but I'll be online to help you with that. Any questions? I'm starting to feel like I'm slipping into a bit of a food coma here. I had a big lunch. I need to get myself some coffee. Okay, so what's next? So essentially what I want from you. When is that due? Oh, cool. Go for it, Angela. You can speak it out if you want. Might be quicker than typing it. Or you can type it. I don't mind.
Oh, can you hear me?
Uh-huh.
Okay. On page 5, the numbers 1 through 8 there, 3 and 4 and 7 and 8. What are those? I mean, they're the low numbers, so does that mean they're the double recombinants? Is there 2 sets?
Oh, okay. I got you. So 1 through 4 are female. 5 through 8 are male. So
Gotcha.
yeah, so essentially that kind of in like Word document format, it's the same format as these data here. It's just these ones are tabulated. So as you can see, we've got 4 categories for each sex. 2 of them are going to be recombinant, which are the smallest numbers, and 2 of them are going to be parental, which are the larger numbers. So to do this chi-square with all 8 categories, you'd have to calculate expected values for each category based on the total, and then calculate your chi-square based on that. Hang on. Yeah, the walkthrough here that we have for the data analysis, this example on page 5, what page is that? 4, all the way going down to page 6 with the odds ratio is an example. You'll need to calculate all of this stuff yourself. Scrolling back up again. For the data that are the start of page 4. These ones just here. I don't know if you can see that. That help? Cool. So you're very welcome. Get your patty. There we go. So, sorry, it smells like one of my daughters is burning something. This is today. I would like to see that Excel spreadsheet. It's not due at the beginning of the next lab. That one is going to be due on Thursday evening, right? And we'll go over that on Tuesday again, just in case you have any questions. So have a watch that recording. I don't think I can watch it with you because I don't think the sound comes through on WebEx for that. But watch the recording that's linked in here. This one here, this Blackboard Collaborate recording. And then try and follow the instructions in that to generate that Excel graph, right? Make it look pretty. So I should get rid of all these beginning next lab because that just confuses things. So this graph is due, hang on, let's write this out here. And there's a mini lab report, which I'll get to in a second. What date is that? That'll be, yeah, that's the one. Oh, hang on. Yeah, that's right. Oh, sorry, that's easy. I can get that date right. The Americans putting the date the wrong way around. Yeah, so Excel graph on the 2nd of July and the mini lab report the following week on the 7th of July. And so that should give you a little bit longer over the weekend to write that up. Okay. So, mini lab report, I don't want this to be onerous, right? That's not the intent, but I do want you to get how to format a formal lab report and what needs to go into it. Okay, so basically I've laid it out here. The main thing that I want from you are the results, right? That's the important stuff. You need to have your Excel graph, which you will already have done for the previous assignment, and a figure legend. A figure legend will explain what that graph shows, so you can have just the figure legend and the figure, and it will make sense, right? It's like a standalone deal. We can talk about that some more next week. You need your chi-squared, hang on, got more fingers coming up, chi-squared test with the chi-square value, degrees of freedom, and p-value, right, for the data that I have given you. You need your odds ratio. You need your, hang on, I think I put up too many fingers there. Anyway, you need your map distance in centimorgans, and you need a short amount of text describing the above, right? So when you write a lab report, you can't just give your results. You also have to describe them a little bit so that you're not relying on the reader to be able to interpret your images as you intend them to, right? So you actually need to walk them through. Like, what does the graph show? You need to basically describe the chi-squared test and the odds ratio and the distance. You don't have to interpret it, right? That's discussion type stuff, but you do have to describe it, right? And so in a sense, in this kind of like short mini format, the figure legend and the results text feels a little bit duplicative, right? You're doing the same thing over. Don't worry about that. That's simply because you have one graph to describe. If you had 4 or 5 or 6, it wouldn't feel so odd. So around those results, you need a short introduction. What are you studying? Drosophila. What's the kind of cross you're doing, right? About that much writing, give or take. Don't bust yourself up overdoing it, right? Just needs to be a basic background so anyone that picks it up can go, oh, right, they're doing a lab report on linkage and Drosophila and blah, right? Very brief methods, right? This doesn't have to be a copy and paste, right? That's not the intent. Just an overview of how you asked this question. And then finish all of that up with a short discussion. What did you find? What does it mean? Okay, so overall, my hope is that this should be about 2 pages long, plus maybe a little bit extra for the graph, maybe, right? Doesn't have to be. I'm not grading you on the length of your report. As long as you hit all of those different things, then you get the full grade, right? But you do need, your full figure, right, with labeled axes, the different stuff going on there. Bloody cat fur. And a figure legend. You don't have a figure legend, you're not going to get full points for your results. You need all of the other things and the text that describes them. That on its own is going to be at least half of your grade for this lab report, which I think is 200 points out of the total for the lab. So it's worth doing well. And then the others, as long as you cover the basics, you're good, right? I'm not going to be penalizing you on missing out one thing or another, as long as you cover the basics. The results, though, you have to have everything, right? So if you want to check any of that with me prior to submission, that's also cool. I'm happy to give it a quick look over. I'm not going to do a full grading because, I'm going to do that anyway. But I'll be able to let you know if you're missing anything. And that's it. And so both those Excel files and the Word files need to be sent to me electronically by email. And please put your name in the file name. It makes my life so much easier. And that's pretty much it. I don't really have anything else to talk about, from my end at least. Does anybody have any questions? And don't forget, we also have time next week to go over stuff in detail if necessary. Questions, questions, questions? No? Awesome. Well, that's thankfully a fairly quick one today. It's been a bit of a heavy morning. Yes, the Excel spreadsheet needs to be emailed to me personally. Actually, you should know what my email address is. But that's what it is. So just email it to me as attachment. Save it with your name in there, right? So Angela's would be Roth Excel file or Angela Excel file or whatever. It doesn't really matter as long as it has your name in there. And that just makes it easier for me to kind of give the right person the grade, right? I don't want to get it wrong. Yes, you email both. That is correct. I think that's it. Cool beans. If you have any questions, again, go through this yourself. And we'll be going through any of the Excel stuff and, whatever else is necessary next Tuesday. All right. Thanks very much, everybody. Have a lovely afternoon. Oh, yeah, that's right. It's the weekend coming up. That's funny. I kind of forget that there are weekends anymore, to be honest. Anyway, have a great time, everybody. See you all soon.